So this is just a brief introduction to homeostasis and also having a look at temperature regulation. So as you already know from GCSE, homeostasis is the maintenance of a constant internal environment, or the maintenance of an internal environment within restricted limits. So some of the things inside our body that we need to maintain within these restricted limits are temperature, blood pH, and let's just have a quick think about the things that can affect blood pH. So when we're talking about blood pH, we're talking about things like the presence of carbonic acid in our blood, or the presence of lactic acid in our blood, which would lower the pH. Water, which would affect the osmotic potential, the water potential of the cells, blood sugar levels, respiratory gases, and blood pressure, and a bunch of other things. Okay, so homeostasis is the maintenance of an internal environment within restricted limits. Now, we tend to do this using something called negative feedback. Negative feedback is where any deviation from a set level or from an optimum leads to a response which returns that thing to the optimum. So for instance, if your temperature rises, that's the stimulus, the temperature rises, this will be detected by a receptor in your body. You will then coordinate a response. In this case, that could be, sweating, for instance, and the evaporation of the sweat would cool you back down to your optimum temperature. And if you go below your optimum temperature, this will be detected. A coordinator would determine a response, e.g. shivering, and then you would return back to your optimum temperature. So negative feedback works in a loop. So it's where any change in the system occurs. So there's a stimulus, and then that change is detected by a receptor. And then you have a coordinator. This is usually within the CNS. And you have an effector. This is usually a muscle or a gland. And then you have a response. So in negative feedback, any change or fluctuation is fed back, and there's a response which reduces this fluctuation. Positive feedback is when the stimulus causes a physiological response to remain on or to increase the change. So instead of reducing the change, it increases the change. These are much rarer within healthy systems. But one common example is that occurs during childbirth. So when you go into labour, you release a chemical, you release a hormone called oxytocin. Oxytocin causes the uterus to contract. When the uterus contracts, the baby's head pushes against the cervix. This stretches the cervix. Stretching the cervix causes nerve impulses to be sent to the brain, that then stimulates the pituitary gland to release more oxytocin, which causes stronger contractions, which causes the baby's head to push more strongly against the cervix, stretching the cervical wall more, sending a greater frequency of nerve impulses to the brain to release more oxytocin. So this is a positive feedback loop. Smaller contractions lead to stronger contractions during labour. Or I really like this one as another good example. The number of cattle running increases, and so the overall level of panic increases, and so the number of cattle running increases again, and so on and so on. I quite like that one as a good example. So two key terms there, positive feedback and negative feedback, that you need to know. And just think about some of the other words that we used there as well. So we talked about receptors, we talked about coordinators, talked about effectors, and response. So in this topic, we're going to look at temperature, blood glucose, and osmoregulation. But let's just have a look at temperature regulation first. So this is a really lovely diagram that sort of explains everything. So let's just have a look at it. The stimulus is increased body temperature. So it could be because you're doing exercise, and you know that when you exercise, you do more respiration. You know that heat is a waste product of aerobic respiration. So heat is given off during respiration. You get hotter. So your body temperature gets higher. Now, your hypothalamus is the area of your body that measures, that detects your body temperature. So when your hypothalamus notices that your core body temperature has risen, it activates the heat loss center in your hypothalamus. This triggers a number of things to occur. So let's just talk about them in turn. One is that your sweat glands are activated. These cause you to secrete sweat. And what cools you down about sweat? When you sweat, it is the evaporation of sweat from your body that cools you down. So the evaporation of sweat cools you down. So the evaporation takes heat energy away from your body, lowering your overall average body temperature. The second is that blood vessels in your skin dilate. Capillaries become flushed with warm blood and heat radiates away from the surface. So it's the radiation of heat away from the surface of your skin that cools you down here. So I just want you to talk to you about this a little bit here. So if we think about blood flowing through your body, we know that blood flows through arteries and then it flows through arterioles and then it's going to head into capillary beds. So we talk about blood vessels dilating. What we're referring to here is the arterioles. So within the arterial walls there are smooth muscles called sphincter muscles. When if we imagine there's there could be a capillary bed up close towards the surface of the skin. So your skin's here, and there could be a capillary bed closer towards the core. So where's the blood going to flow? Okay, is it going to flow up here, or is it going to flow down here? So if you are hot, you want it to flow closer to the surface of your skin. So the sphincter muscle in your arteriole relaxes. When that relaxes, this widens the lumen of your arteriole, causes the arteriole to dilate, and so blood now flows through this arteriole into the capillary. So more blood is flowing up here in comparison to flowing down here. So it's not the capillaries that dilate, it's the arterioles that dilate, allowing the blood to flow into the capillary beds closer to the surface of your skin, and then the heat radiates away. So these things cool your body down. Your body temperature decreases, blood temperature lowers, and your hypothalamic heat loss center shuts off. You return to your original body temperature. If the stimulus is decreased body temperature, so your blood is now cooler than the set hypothalamic point, this activates the heat promoting center in the hypothalamus, and it causes skeletal muscles to become activated. They shiver. This means they do more aerobic respiration. The aerobic respiration releases heat as a waste product. This heat is given off and it warms you up. Now, the skin blood vessels constrict. Now, just make sure you're taking note of this term, constrict. Now, the blood vessels constrict because the sphincter muscles in the walls of the arterioles contract. Okay, remember, it's muscles that contract and relax, but the blood vessels constrict. So blood is now diverted away from skin capillaries and goes into capillary beds that are closer down to the core of your body. Less blood goes towards your skin, and this minimizes radiation of heat away from your body, and so this raises your body temperature back up to the optimum. So you've got this image on the sheet, your handout, but I want you to just take a minute to have a look at it, check that you're happy with all of it. So if we just think about what's going on in your hypothalamus, so your hypothalamus is measuring your core temperature, your blood temperature, not really the temperature of your skin. And it's the thermoregulatory center in your hypothalamus that's measuring this. So on the diagram here, I know it says effector nerves, but we know these as motor neurons. Okay, so motor neurons are sending action potentials, so motor neurons are then propagating action potentials down towards effectors, such as the arterioles in the skin, which we mentioned earlier, the hair erector muscles. So we know that when we are cold, the erector muscles in our skin contract. And when they contract, they cause the hairs to stand on end. This traps a layer of still air and it insulates us. Students sometimes write that it keeps us warm or it makes us warm. It's not doing that. It's not heating us up. What it's doing is it's trapping the body heat that we are producing. So it insulates, and that's a key term there. And remember, sweat, it's the evaporation of sweat that cools us down. And skeletal muscle, this is all to do with increasing respiratory rate. Adrenaline, for the release of glucose, for all that extra respiration. And thyroxine to increase our metabolic rate, which is all linked to the same thing. So this is on your sheet, but I just want you to take a minute to read through each of these in turn. So response if the body temperature is too high, and response if the body temperature is too low, and see if you've got any questions about it. Most of these should be pretty straightforward, but actually this word we haven't mentioned. We've talked about the movement of blood, but we haven't mentioned this word vasodilation and vasoconstriction. So make sure you're comfortable with those two terms. And remember, the reason you decrease your metabolic rate when you're too hot is because respiration is exothermic. It gives off heat as a waste product. And then we didn't mention also behavioral changes. So what you might do. So take a minute to get your head around those. So if you get sick, the set point in your hypothalamus changes. It goes higher. So even when you are at what should be regarded as your normal optimum body temperature, you feel cold. And the reason for that is your body is trying to raise your body temperature to provide a hostile environment for these pathogens, and also because of your higher metabolic rate to allow all the things in your immune system to work correctly. But if you go too hot, if you go to about 41 or above, this becomes really dangerous because you lose regulatory control. So basically this section of your hypothalamus stops working and you lead to positive feedback. So you just get hotter and hotter and hotter. And this can become really dangerous, which is why when you're sick, the doctors monitor your body temperature and they might have to do things actively to cool you down. So let's have a look at these two little guys. So over here, we've got a fennec fox, and here we've got an Arctic fox. And here we're looking at physiological adaptations to temperature. So we can see here that the fennec fox, he has these lovely, beautiful ears. So he has a large surface area to volume ratio, and you can see in his ears all the blood vessels are really close to the surface of the skin. This allows heat to radiate away from the body. So we've got an increased surface area to volume ratio, and this allows heat to radiate away. It reduces the distance for heat to travel, keeps him nice and cool. And then this Arctic fox, he's got these lovely little ears. He's also got lots of nice thick fur. He's got a lower surface area to volume ratio, and it's reducing the radiation of heat away from the body. So let's have a look at these guys. So it might be common practice for people to say that these are cold-blooded animals. They're not cold-blooded. Their blood is not cold. Their blood is just the same temperature as the external environment. So if the external environment is cold, they're cold. If the external environment is hot, they're hot. So what is the correct term? The correct term is ectotherms. They're not cold-blooded. Their body temperature just changes with the external environment. So they cannot increase their rate of respiration. And I want to clarify this. That doesn't mean they can't ever increase their rate of respiration. They just cannot increase their rate of respiration as a method to control their body temperature. Whereas we are endotherms. We can maintain our body temperature within strict limits. So we do these things like sweating, or like shivering. Now, ectotherms, they aren't powerless. They have other ways of keeping themselves at the correct temperature. So they are more likely to show behavioral adaptations like basking or burrowing and seeking shade in order to maintain their body temperature. So just a quick recap. Negative feedback is when the stimulus causes the corrective measure to be turned off to reduce change. whereas positive feedback is when the stimulus causes the physiological response to remain on to increase change. I'm also going to talk to you very briefly about something called the secondary messenger model. So basically, there are two types of hormones in your body. There are lipid-based hormones and there are protein-based hormones. Now, lipid-based hormones can just move directly through the phospholipid bilayer. They're lipid-soluble. Now, these can move directly into the cytoplasm and they can have their effects directly on the gene or the enzyme or whatever it is inside the cell that they want to have their effect on. Great. Primary, they have their effect directly. However, most hormones are proteins. That means they cannot move directly across the phospholipid bilayer. So instead, they need to bind onto receptors, complementary receptors on the cell surface membrane. So this enables protein-based hormones to be more specific because those receptors might only be present on certain cells. We call these cells, tissues, or organs target cells or target organs because they're the only ones that can interact with this particular hormone because they're the only ones with the receptors on their cell surface membranes. So this means that when the protein binds onto the receptor, it has to trigger something to happen inside the cell. So it has its effect sort of from a distance. It still remains outside, but the effect that it's causing is inside the cell. That's what the secondary messenger model means. You need to know about an example of a secondary messenger, and this is adrenaline. So I'm just going to talk you through adrenaline as an example. So let's have a look here. So this is our cell surface membrane here. And where we are here is in the liver cell. And we've got this transmembrane protein that is the receptor for adrenaline. So we can see this transmembrane protein, that is the receptor for adrenaline. And it has this specific tertiary structure that's only complementary to adrenaline. So adrenaline comes and binds on, and we can see that when it binds on, it causes this protein here, which is adenyl cyclase, you need to know that name, to change shape. Can you see how it's changed its shape here? It's changed it from being in its inactive version to being in its active version. So now adenyl cyclase has been activated. So what happens next? The adenyl cyclase converts ATP into cyclic AMP. So now we've produced cyclic AMP. The second enzyme that you need to know about is an enzyme called protein kinase. It sits in its inactive version until it comes into contact with cyclic AMP. Cyclic AMP activates protein kinase. So now we've got active protein kinase. Protein kinase then converts glycogen into glucose, and then that glucose can then be released into the bloodstream. To increase the blood glucose levels, and that's how adrenaline increases your blood glucose levels. So let's just talk that through again. The adrenaline, which is complementary, so it has this receptor site on this transmembrane protein, binds on and causes a change in the shape of adenyl cyclase, activating it. So now you've got active adenyl cyclase. This converts ATP into cyclic AMP. So now you've got cyclic AMP, which activates protein kinase to then start converting glycogen into glucose. This is a cascade reaction. So you need to know this process: adrenaline, adenyl cyclase, cyclic AMP, protein kinase. Okay, that's everything that you need to know. You should now be in a position where you should be able to tackle those thermoregulation questions. Well done, and thanks for listening.